Beautiful. So we're just going to briefly pause to talk about DNA. And I specifically like to do this because normally in the 3U, I don't really talk about DNA structure too much because that's in the 4U. But this is split class. And we also have people who are in the 3C biology. And that doesn't lead to a next class. So I really like to make sure that those students get a sense of how DNA is actually structured and what it actually looks like. So this kind of bridges those two curricula. So we'll take a look. So first I will, of course, write the title again, handwritten, so I can be cool like you guys. Sorry? Yeah, no, my projector's acting up. I'm going to have to replace it, and it's kind of flickering today. It's being annoying, so sorry about that. Sure, yeah, we won't be spooked out. Speak now or forever hold your peace if you're afraid of the dark. Okay. All right. That's better? Wow, I find this sort of gloomy and depressing, but okay.
That's how we like it.
Don't tell me what to do. Deal. Yeah, I'll go check in a moment. What I want to start you with is this. Recall that genetic information is stored in the DNA found in the nucleus of a cell. And obviously, when I say the cell's nucleus, what kinds of cells am I talking about?
Eukaryotic.
Eukaryotic cells. Where would it be found in a prokaryotic cell?
Cytoplasm.
Just in the cytoplasm. Very good. Well, you guys there's some paper here if you need it. Walton, I'll get you some. And I'll freeze it. The beef. Sorry? What? Are you talking to me?
I thought you said what? Sorry.
Oh, no, that's okay. I did say what. But were you trying to speak to me?
I was talking as well, but they said sorry.
Oh, okay. Okay, sorry. I apologize. So, for example, let's look at an animal cell. And if that is the cell membrane, we'd expect to see all sorts of different organelles in there, but I'm going to focus on this, which is the nucleus. If that's the nucleus altogether, what do we call the actual boundary, the actual structure? Yep, specifically the... Yeah, very good. Well done, guys. The nuclear membrane.
That's an upshot, or that's a reason for the endosymbiosis theory.
Yeah, that's right. Yep, exactly right. Man, you guys are good. And then inside it, we have all kinds of this material that we call DNA. Right? What's DNA? Yeah, that's what the DNA contains, absolutely. The DNA contains all the information that makes up your genome. It contains all of your genes. So all of your traits are determined by the information stored on your DNA. What's DNA? Yep, absolutely. What is it? That's what it contains. That's what the information on it is. What's the DNA? That's how the information is stored on. And these aren't bad answers, by the way. These are all good answers. But what is DNA? It'd be like me saying, what's a book? And you're saying, well, it tells stories. True. 100% true. What's a book? Well, it's got words. Absolutely true. You just went down to letters. Well, the words are made of letters. Totally true. What's a book? Well, a book is a bunch of pages of paper held together in a bounding, right? What's DNA? You've told me what's on it. It's instructions. That instruction's stored in a code. The DNA itself. Yeah, it's the storage medium. Absolutely. What is it? Like, is it a book? Is there like a little book in there and like little cell elves go and open it up and they read it and they go, aha, this is how we make the mitochondria. And then they go do it? Yeah. Correct. And that's what I'm about to teach you. All about, no, it's not correct. That's incorrect, team. What is correct? Yeah.
It's the blueprint of the cell.
Yep. But I want to dig in what the actual thing is. Because you're right. I'm not criticizing. These are all good answers. What do you think it is? It's got to be something like a physical, tangible thing, right? Like when you say a blueprint, like that both refers to like the concept of like a drawing, but it also refers to a piece of paper that's blue with white lines on it. Is there a piece of paper that's blue with white lines on it in the cell? No. So what physically is this stuff?
It's like a physical filament almost?
Yes! That's an excellent answer. It is. It's a big, long chain. It's a filament. It's a molecule. It's a big, long molecule. One big, long chain. Actually, in the human cell, 46 big, long chains. Your genome is cut up into 46 pieces. Just like an encyclopedia used to be cut up into separate books, so that's easier to kind of find the information. Your DNA is cut into smaller pieces for the cell to access it. But DNA itself is a molecule. And specifically, it's a polymer. Filament is another good term for that. It's a chain. It's a big, long thing put together. So first and foremost, it wouldn't hurt to know what DNA stands for. Does anyone know what DNA stands for?
This speaker brings it up all the time. Something about acid. I know that's what the A. Or
You're right. The A stands for acid. Yeah. Is that a hint? No, we're just chilling. Okay. Oh, sure. Yeah, you bet. Huh? DNA in the cell. DNA and the cell. So DNA stands for deoxyribonucleic acid. It occurs to me I capitalized the wrong letter. Sorry. The whole point was to show you where the acronym comes from, and then I capitalized. Damn it. Okay, there you go. So, no. I'm just showing you here. Deoxyribonucleic acid. It's not a real name, so it would just be lowercase letters, right? Like it's like you don't call your pencil, pencil, and write it with an uppercase P. It's a pencil. This is DNA. But I'm showing you where the letters come from. Deoxyribonucleic acid. By the way, what do you suppose RNA is? Nope. It's a good guess, though. No, it's not reoxy. What do you suppose it is? Deoxy just means D means taken away. Oxy means oxygen. So deoxyribonucleic acid is actually just ribonucleic acid that's had an oxygen taken away from it. So RNA is just... And why I accidentally capitalized the R before? Ribonucleic acid. DNA and RNA are almost the same molecule. We'll talk about the ways in which they are different, but that's kind of the first thing to note. We said, remember that our cells use DNA as the molecule in which they store their information, but prokaryotic cells, some use DNA, but some are still using RNA. They're very similar molecules. They do the same job. Yeah.
Is one better?
We're going to talk about that. It's a good question. We're going to talk about that. So I'm going to kick the can on that. This molecule is a long chain of individual nucleotides, which is just a small molecule that can be put together to make a big one. All right? So I'll give you a minute to write that, and we're going to draw it. And to be clear, this is actually jumping into grade 12 biology for those of you in the U stream. In the C stream, this actually belongs to your curriculum here, but it's actually good for both groups to know it. I actually think it's a good idea to teach this in the grade 11 anyway. It's kind of nice to know what you're talking about when you're talking about DNA. What is it? What is this stuff? Does that make sense? So the basic idea is this. Ready? If you take a cell and there's the nucleus and here's all the DNA, if we zoom in on the DNA, DNA is a molecule with a famous shape. You've probably seen it before. It's got this double helix shape to it. Have you seen that before? They usually kind of draw it like that. Does that look familiar? Okay, so DNA has a double helix shape. Helix just means it twirls around, and double helix means this is almost like a spiral staircase. If you picture the two banisters of a spiral staircase, do you kind of know what I'm talking about? So it's got a double helix shape. And then, if you actually look at this, each piece of DNA is just one of these little sections here. I'm going to try to differentiate between them. You kind of see how I'm highlighting each of these individually? Each section is just one little component like that. I guess this next one could look like that. So those are the little building blocks of DNA. Does that kind of make sense? And if we were to zoom in on just one of those, we would see that DNA is made of individual nucleotides. And a single nucleotide has 3 parts to it. All right? And again, this is a little bit more than you need to know, but it's not a big deal. It's very doable. It's got a phosphate group, which we put there. It's got a sugar. I've already drawn this poorly. It's got a phosphate group, which is connected to a sugar, which is connected to a base.
Is there any particular reason why the sugar is a house?
Yeah, because it's the molecule is actually shaped like that. The sugar deoxyribose is a type of sugar molecule, and if you actually look up the structure of it, whoa, going to mole view, team. We're going to mole view. It's my favorite place to be. Now watch, it's going to give me the linear shame, and I'm going to be so not annoyed. So if you look here, this is what we just talked about. That is the sugar part there. That's the sugar part. This part here is the base. Does that make sense? Can I get this without the uracil attached? Because we don't want that. Yeah. So that is the sugar right there. And do you see how it has that kind of pentagon shape? Yeah. Obviously, it's got more than the pentagon going on, but that is the sugar part. And then I can show you what the phosphate looks like. It looks like that. That's a phosphate. And then I could show you, oh golly. And then I could show you one of the nitrogenous bases. There's a bunch of the different ones, but I could show you this is one of the bases. This is a base called cysteine. This is another one called guanine. Right? Yeah. Adenine. So those are what the bases look like. You don't need to know that. All right? You do not need to know that. But when we draw it, just to show you, we draw it just as like a simple line drawing just to make life easy, but those are what the parts actually looks like. So if you put them all together, thymine nucleotide. I wonder if I can just say nucleotide and it'll show me one all together. Yeah, there it is. So this is what it actually looks like. There we have the phosphate group. Can you see that? So this is kind of rotated to our picture, but there's the phosphate group. there's the sugar, and then there's the base. So if you look at it this way, there's the phosphate group, there's the sugar, and there's the base. You do not need to know about this in 3 dimensions. And can you see how in 3 dimensions it's a little bit clumsy, but it basically lines up kind of like that? Right? If you look, phosphate, sugar, base, phosphate, sugar, base. So that's why we draw it that way. Does that make sense? Now imagine if you put these end to end, you can make a molecule. So if we want to imagine that, all right, so again, we'll draw this just a little bit bigger so we can label each part. This is a phosphate group, which is a PO4 group, if you're wondering. This is a sugar. Specifically, it's a deoxyribose sugar, unless it's RNA, in which case it's a ribose sugar. That's the only difference. So in this case, because we're looking at DNA, we'll call this deoxyribose, which has a formula of C6 or C5. What's the formula for deoxyribose? I should know this. You don't need to know this. C5H10O4. Should have known that. Yeah, but C5H10O4, you do not need to know that. You don't need to know either of those things. I'm just putting it there so you have a sense. Does that make sense? And then here, I can only write base because this is always the same. This is always the same. But the base is variable. There are 4 different bases. And the reason there's 4 different bases is how the code is going to get stored. Your DNA is like a book if instead of a book being pages, a book was just a long string of letters, almost like a scroll. You've seen scrolls in the past, right? The idea of like a piece of paper that just keeps going. Now imagine that the scroll is so thin it can only hold 1 letter at a time. Well, actually 2, because it's a double helix, right? So it's 2 chains there. So imagine that scroll could only have 2 letters, then 2 more letters below it, then 2 more letters below it, and so on and so forth. That's basically what your DNA is. Just imagine a scroll, but the scroll is so narrow that it can only hold 2 letters in a row, and then you have to go to the next row and the next row. And imagine that the alphabet doesn't have, oh God, I'm going to have a moment here. Our alphabet has 26 letters. Look at me knowing how many letters are in the alphabet. I can even say them in order, team, if I sing. No. Oh God, I would not torture you, I promise. I mean, at some point I will probably sing for you and you'll regret it. But in the meantime, I won't torture you yet. So you don't have to draw this. Everyone, look at me. Look at me. Look at me. Look at me. Don't draw this. Right? We all promise not to draw this. Okay. You can't have already drawn it because I haven't drawn it yet. Now let's focus. Okay. So imagine we have a scroll. Oh, look, it's a scroll. It's a scroll. Whoa. Okay. There it is. It's so scroll looking. Alex, how am I doing? Just say great. Just say great. Shut up. Excellent. Thank you. I appreciate it. And then imagine the alphabet stopped at D. That's 4 letters, right? A, B, C, D. Okay. So now imagine it just went like, I don't know, A, C, D, C, B. I can do whatever I want. I'm spelling my own words, Kate Odette. C, B, A, B, C, A, C. This is like your DNA. Does that make sense? You can only read it by reading down because there's one thing there. And then there's actually a whole separate story written on the other side. Does that make sense? This is one strand, and it's its own story. It gets read by itself. And then over here, there's actually a totally different strand, and it gets read by itself. The 2, as it turns out, they're related to each other. We'll talk about that later. You don't have to worry about it. But you read just one column. Does that make sense? That's how you have to picture your DNA. It's a big long chain of these put together. Does that make sense? So I'm going to delete that for now because instead of talking about a scroll, we're just going to talk about DNA. So we can imagine this whole thing is called a nucleotide. A phosphate, a sugar, and a base make a nucleotide. So there are 4 bases, and therefore 4 possible nucleotides. The first we short form A, and it's adenine. T is thymine. G is guanine. And C is cytosine. You do not need to have this memorized for this course. You'll need to know this if you take grade 12. But you don't need to know it, though you will. It'll kind of stick in your memory. But the important thing is, this is the alphabet of the genetic code.
Do you teach us in a just 3U class?
Sometimes not. I like to. I think it's helpful to know what you're talking about. I think it's helpful when you hear word DNA to not have it just be like a metaphor. Oh, it's a blueprint. Oh, it's a library. Oh, it's a book or whatever. It's helpful just to know what you're talking about. It's a chain. It's a big chain, and that chain has letters, and they make a code. That's it. Chain has a letter, and it makes a code. And so therefore, when we look at DNA, so I'll give you a minute to write this, I promise. Does it smell like something's burning? Anyone smell that? Am I having a seizure? I don't think I'm having a seizure. I don't know. Oh, you know what? They must be soldering somewhere. That is the smell of solder. Like the flux they use for flux paste when they do like copper soldering, like plumbing. They must be upstairs soldering some pipes. Yeah, that's flux solder for sure. Love that smell. It's a good smell. Ever do any plumbing? It is the most satisfying. Do you know the basic idea of soldering? It's basically you take a pipe, you fit it inside of another pipe. How do you get them to stick together, like a copper pipe into a copper pipe, to stick together so that they are airtight? You put this flux paste between them so that there's this just material that's in there, and then you heat it up with a torch so it gets really hot, and then you bring a metal that melts at a fairly low temperature up to it, called solder, and when you put the metal there, it gets sucked into the gap. So it melts, the metal will melt, and it gets sucked into the gap and takes the place of the solder. The solder will basically boil off, and this metal will take its place, and now when you cool it, it hardens, and now you've got a metal joining the 2 metal pipes. But it's such a cool thing to watch, because you just take this metal wire, and once the fitting is good and hot, you go like this, and it just goes, and kind of sucks in and solidifies, and it's really cool. It always makes me feel very, manly. Yeah, it's a different kind of soldering, but it's the same basic premise. Soldering is like welding, but in welding, the metal you melt is the same as the metal you're joining together. In soldering, you do it at a lower temperature, and you melt a different metal to hold the 2 things together. So soldering, and then brazing is a third one, and I'm not sure what it is. Maybe it's just using brass as the I don't know. Anyway, so soldering, welding, and brazing. Welding for sure is when the metal you are using to join them is the same as the metals you're joining. Soldering is when the metal is different, and brazing exists, and I couldn't tell you what it is. This has been our metallurgy and trades lesson for the day. Now we're going to go back to this. So, one second. So when we examine our DNA, when we look at that, there's our DNA molecule, right? We usually will represent one of the strands. We can actually look at how it's held together. And how it's actually held together, that yellow strand, is it would be phosphate, sugar, base, connected to another phosphate, another sugar, and another base, which is another phosphate connected to another sugar, connected to another base, which is another phosphate connected to another sugar, connected to another base, which is another phosphate connected to another sugar, connected to another base, etc., etc. And each of those bases will either be an A or a T, or a G, or a C. And the order of those, just like the order of letters gives us words, the order of these gives us the code. Does that make sense? So we don't usually go to all the trouble of drawing it like this. Usually we would actually just write that as A, T, G, C, G, T, A, T, T, G, A, C, so on and so forth. Does that make sense? That's how we usually write it. That's all your DNA is. It's a big line of that. And you might think, well, how do you write an interesting story? How do you make an interesting blueprint with only 4 letters? Right? In English, we have 26 letters to work with. In Italian, they have 24. That's still a lot more than 4. On the other hand, when you think about it, your computer has how many letters to work with? 2, 0 or 1. And it does some pretty cool stuff, right? So this is twice as much as a computer. And here's how it works. Here's the trick. So each of these is a single nucleotide. Right? Nucleotides form words in groups of 3. All right? So 3 bases form a single, we use the word codon, but you can basically think of that as a word. Your DNA is coded, and it is a code, it's a language consisting entirely of 3-letter words. Does that make sense? And again, how many letters are there to work with? 4. All right, math whizzes. How many words are possible? You have 4 letters. The words can only be 3 letters long. How many words can there be? 12. Nope. Good thinking. Not 12. You think about it. When you go to make the first letter, how many choices do you have? 3. Not 3. It could be an A, a T, a G, or a C. So the first letter, you have 4 choices. Then when you go to make the second letter, how many choices do you have? 4. Then when you go to make the third letter, how many choices do you have? 4. For a total number of combinations, that is 4 times 4 is?
16.
Times 4 is? 64. Good. So there's 64 possible words. All right? So there's 64 possible words, which is actually more than you need because... Does anyone know what your DNA actually codes for? See, we say that it's blueprints, but what's it blueprints to? Well, it turns out the DNA gets read by the cell as instructions to build proteins. We're not going to go super into detail here. All you need to know is that proteins are the machines of the cell. They're the doing-y bits of the cell.
Can you scroll just a little bit?
Sure. Everything in your cell that does a job is a protein. So your muscles, the part of it that actually moves, that's a protein. But also like all of the machinery that actually reads the DNA, those are all proteins. The machine that replicates the DNA, makes new copies, that's a protein. The machines that form the spindle fibers and pull the cell together, that's a protein. The machine that inside the mitochondrion that breaks sugars apart, those are proteins. The machine that stores that energy in a molecule called ATP, that's a protein. Everything's a protein. Everything in your cells that does something is a protein. That's what cellular machines are made of. And this is the instructions on how to build that protein. And as it turns out, there's only 64 possible words. That's way more than we need because proteins are made of building blocks and there's only 20 building blocks of proteins. So 64 words is actually more than we need. So there's some redundancy there. Does that make sense? Imagine if you went to go to your building kit and all you have to build with is 20 different pieces, right? Imagine a Lego kit that has 20 different pieces. all these instructions say is, cool, these 3 letters, you read them and it says, great, the first piece is this one. Then the next 3 letters say, okay, the next piece is this one. And so the DNA is instructions on how to build proteins. Does that make sense? That's it. And proteins are made of 20 different amino acids put together. So proteins are built from building blocks called amino acids. And again, this is a little bit beyond the scope of this course, so I don't need you to memorize this. I just want you to understand it. I'm so sorry. I feel like we should switch one day.
Blank printer paper at the front.
Yeah, tons.
Need enough for a taco truck logo for everyone.
Also,
I mean, I don't know how many blank pieces of paper are involved in a taco truck logo, but is that plenty?
That's good. And I might
Awesome.
on Thursday ask your class to judge the taco logos. With
God, do we want to do that?
the criteria.
See how quickly they sign up for being
They're not all
judgmental?
Tacos, they're just
Also-
food truck logos.
were you gonna ask Helper for line paper?
I got line paper.
Oh, yes, that's good. I need that too at some point.
I stole it from Reeser.
Oh, well, then I need that.
I was like, why steal from Calford when I can steal from Reeser? There are 20 amino acids, so 64 words is more than enough. Which I actually want to pause here because this actually isn't that far off from there being a little book inside your cell and little elves that read it. It's not a book and they're not elves, but it's pretty darn close. It's basically a scroll. It is basically a big long scroll, and there's little machines that go along and read it. And they read it 3 letters at a time, and those 3 letters tell it, oh, the next building block is this. So when your cell wants to build a new mitochondrion, it literally, I mean, that's a bad example because that's got its own DNA. When your cell wants to build a new ribosome, it goes to the section of your DNA that is for building ribosomes, and it just goes, okay, let's get building. Read these 3 letters, cool, here's the first building block. Shifts along. Oh, these next 3 letters, boom, there's the next building block. Shifts along. The next 3 letters, there's the next building block. It really is that direct. It's a set of instructions of how to build things. Does that make sense? That's not a metaphor. It's not a metaphor. It literally is coded instructions of how to build things. It's actually kind of weird to think your cells actually have their own language. Not a spoken language, but a written language. It's pretty crazy. And not just your cells, but every cell in existence has a written language, has a written code that stores coded information that can be read by the cell to build things. This is why you can take a section of DNA out of, and this has been done, out of a jellyfish that glows green, and you can inject it into the cells of a fetal pig, and literally that fetal pig's skin will start to glow green. Because that jellyfish DNA, that section that you took out, had an instruction on how to build a protein whose only job is to glow green. And if you put that instruction inside the cell doesn't know where it came from, it just reads it. 3 letters, next building block. 3 letters, next building block. And before you know it, it's built GFP, which is called green fluorescing protein. And this is a protein that makes things glow. And this is actually, GFP is like, we use that gene constantly because we use it to say, hey, if we put that gene in this part of the developing fetus, where do those tissues eventually form from? So we can pick an early embryo and say, this thing's only 30 cells big. If we put that protein just into this one cell here, we can see what that cell eventually turns into. But the point being, your cell doesn't think, it just sees the instructions and builds whatever the instructions say to build. Does that make sense? And if you change the blueprint, the cell will change how it builds things. This is what a mutation is. That's what we're going to learn later on. All a mutation is just one of those letters. A G gets replaced with a C, or a C gets replaced with a T, or a T gets replaced with an A. Whatever it is, that's all a mutation is. It's just changing one letter, maybe more than one letter, and that's going to change what the thing the cell builds is. And sometimes that change makes no difference. Sometimes it makes a huge difference and kills the organism. Sometimes it makes a difference and doesn't kill the organism. And that's what we call a mutation. Does that kind of make sense? That's a lot. Like, that's a lot. This is esoteric stuff. Again, it's kind of borrowing from the 4U course, and I don't want to go over and above your head, but I think it's helpful to understand that. So we're not talking about DNA as an abstraction anymore. DNA isn't just this curly-looking stuff inside of a cell. DNA is a molecule, and if you look at that curly-looking stuff, we actually are able to figure out the shape of that molecule, the structure of it. And viewed from zoomed out, it takes this double helix shape. It's actually two scrolls of information stuck to each other. Each of those scrolls is made up of individual chunks, individual pieces. We call those individual pieces nucleotides. And a nucleotide itself has three parts. It's a phosphate, a sugar, and then this base. And the base can change. There isn't one base. There's only one sugar we use, either ribose or deoxyribose. There's only one type of phosphate we use. It's always a phosphate. But the base actually doesn't have one type. It has four types. And the identity of that base is going to serve as the source of coding. That's where the information goes. Because just like having an A, a B, a C, and a D lets you write such amazing words as dad. Oh, look, it's my dad. Bad. Oh, no, my dad is bad. Cad. The reason he's bad is that he's a cad. I want to get away from him, so I'm going to call a cab. I don't know. The truth is, 4 letters doesn't give us a lot of options in English, but it gives you plenty of options if all you need is to control which of 20 building blocks to put together. Does that make sense? Coded information.
What's a cat?
A cat is like a person of low morals.
Also, it's kind of a
Which can I tell you the funniest Jeopardy moment I've ever seen? The clue was a gardening utensil or a person of low morals, and the answer they were looking for was rake. But obviously, what was the answer the first person gave? Right, of course, right? Like, obviously, that's what they were going to say, and I thought that was really funny. And then they said they were wrong, and I'm like, how can you call that wrong? That's clearly a fair answer to that clue. Anyway. Right. Anyway, that's the end of my story. Sorry I interrupted you.
It's okay. When you said that you can implant jellyfish that glow and to make things glow.
Yeah? Do you want to see?
Is that why the boy in the Lorax glows?
Yes, that documentary about real life, the Lorax, that's what you interrupted me to ask.
Maybe.
I'm just checking. Okay, carry on. How am I doing at being more sensitive, guys? Good? Really good? Okay. Yeah, so just to show you, like, this has been legitimately done. I want to find one that's a real photo, as opposed to just a fake one. Because those are fake. Like, those are fake. But that is not faked. This is sort of enhanced. But literally, you can make animals that their skin glows in the dark because they've been injected with GFP. And GFP is just green fluorescing protein. Well, it's not a question of being injected with it, because again, they don't put the protein into the mice. It's weirder than that. They go into the mice's cell when it's just an embryo, and they put the DNA in. So we're not pumping green fluorescing protein into the animal. It's more interesting than that. We're putting the DNA for how to build that protein into it, and then the mouse just builds it itself. So we use green fluorescing protein all the time. It's a really important molecule in biomedical research, like cell biology. And that's what that molecule looks like. It's a tremendously complicated molecule. Do we have a space filling model? Yeah, it looks like, that's useless. It looks like that. So it's a big molecule. Lots of atoms put together. And it has the character that it glows green. We also have a blue fluorescing protein. I'm not sure if there's more than that. This was like cutting edge stuff when I was in university, and now it's like old hat. It's been around forever. But it's a tool that still gets used a lot. Okay, so why do we learn all this? Because what it means... Oh, yeah. Oh, there's more, of course. Genes, alleles, and DNA. A gene, therefore, is just a section of a DNA molecule that codes for a protein. The action of that protein, what that protein does, is what actually gives you the trait. So like when we were talking about pea plants and we were talking about their genes, there's some gene that produces a protein. And one version of that protein results in making a plant that grows tall. And a different version of that protein results in a plant that grows short. But the actual doing bit is the protein. The gene is just the instructions to build that protein. So then different alleles have different base sequences and so produce different proteins. Sometimes mildly different and sometimes very different. So for example, skin tone is actually controlled by a few genes, not just one. But basically, the proteins that are responsible for producing melanin, for producing pigment, there's some alleles that make proteins that do that very effectively, and so they produce a lot of melanin. And then there's some genes that have slightly different versions of the gene, and those proteins do it less effectively, and they produce less melanin. Some very ineffectively, and your body produces very little melanin, and that results in us having different skin tones. But do you see how the gene is responsible for building a protein that actually does a thing? Does that kind of make sense? It's really helpful to kind of understand the action of these things. So let's imagine then, gene IMAG. Genes often have like little 3 or 4 letter codes. So we're going to go IMAG for the imaginary gene we're looking at.
Is I even an option?
With 2, we're just giving it a name, a name for us. So we could imagine a gene with 2 alleles, right? The first allele could have the code, I don't know, GCCGACTTGCATAAGC. I don't know, I'm just making stuff up here. And that would obviously keep going because a typical gene will be anywhere between 50,000 and 5 million base pairs long. So, this one, not very big. And then the second allele might be GCCGACTTG. And then here could be where the change is. GCGCATG. Does that make sense? So there could just be a section that is different between the 2 alleles. And that's what would give us the different trait. And sometimes the alleles are quite similar, and sometimes the alleles are quite different from each other. Does that make sense? So the different sequences result in different alleles. By the way, you probably have done something like this in your life, where imagine if you're a guy who builds like blueprints on how to build things, it probably wouldn't be that uncommon for you to cut and paste an old one and then just change it, right? Can you imagine that? So say like you have a blueprint for how to build a house, and then somebody else orders a different house, and it's pretty much the same, but they want a bigger kitchen and a smaller living room. Rather than start from scratch, you could imagine taking the other one and then just changing it a bit, right? We'll move this wall here, we'll put a doorway there, rather than just starting from scratch. You can kind of think of this that way. They're mostly the same, but small differences would make them a little bit different. Big differences would make them very different. Does that feel okay? Again, this is esoteric stuff. So if it feels a little confusing, please don't be discouraged by that. It takes some time to wrap your head around this. I think it takes time to wrap. Maybe your head is already thoroughly wrapped around it and you're saying, God, why does Orlando keep acting like this is challenging? But I find it challenging. Questions about that? All right, one more thing and then I'll be quiet. And then you'll have some time to get some work done. The last thing of note is momenti diploidi, which is to say, don't forget we're diploid.
Was there a 1 and 2? There wasn't a 1 and 2.
Well, there kind of was, but okay, we'll do away with the numbering system. In my mind, there was a 1 and 2, and now this is the third. There was a first subheading and a second subheading, making this the third subheading. In the first half of the class, we did one thing. Then the second half, we did this. Now we're on to the third half of the class, where we'll discuss this. This is as many halves as I want there to be. Allow me to demonstrate. 1 half, a second half, a third half.
That goes into more than one whole, which would be
Sure.
into fourth period.
Sure. I'm okay with that. What do you guys got? Fourth period. Ooh, math. Ooh, that's important.
Spare.
Oh, music. Spare. Anyone cares about these things? Listen, science is serious business. Right? This isn't math class. Math is for fun. This isn't for fun. This isn't English class. We're not here to better ourselves. This isn't history. We're not here to catch up on our sleep. This isn't geography. We're not here to pick up girls. This is science class. This is serious business. End of rant. Okay. I apologize to all English, history, math, and geography teachers I may have offended. Yes. Always. Always recorded. So anyway, don't forget that we're diploid. What does that mean?
Two. Dies 2.
Yeah, very good. So, sexual organisms have 2 copies of each and every gene. Asterisks, except for sex-linked genes, but we're going to ignore that for the time being. All right, so the X chromosome is the one exception, where if you're chromosomally male, then you only have 1 copy of the X chromosome, in which case you'd have 2 copies of each and every gene, except for the genes on your X chromosome. Is everyone cool if I don't write that for now? We're going to do a whole note on sex chromosomes. Everyone's chill if we just ignore that for the moment. So I want you to accept that this is a slight oversimplification, but that's okay. Okay, moving on. So sexual organisms have 2 copies of each and every gene. Genes are found on individual strands of DNA called chromosomes. So the entire genome isn't stored on a single piece of DNA. As it happens, yours is stored on, we said already, does anyone know how many chromosomes humans have? 46. 46, which is actually 2 sets of chromosomes, because you get 1 set of chromosomes from your biological mother, and you get 1 set of chromosomes from your biological father. So we'll speak here for humans, but be aware that it's a different number for different organisms. For humans, there are 23 unique chromosomes. Again, big asterisk, 24 if you count the Y chromosome, which for now we're going to ignore. The 23 unique, we're going to pretend we're all female for now, which we all start out female anyway, and then we'll get to that. So, for humans, the 23 unique chromosomes.
So,
Yep.
females are dominant, and then males are.
It's not that simple. We'll get there. Gender, not gender. Gender is a whole different thing. Sex and the determinism from that really varies from animal to animal. So in humans, it's controlled by a chromosome mostly, though there are other factors. But that's not true for every animal. A lot of reptiles, whether you're male or female, depends on nothing but what the temperature of your egg was at gestation. And animals at a warmer egg gestation temperature will end up being male, and at cooler temperatures will be female. Some animals are actually born female, and then some of them transition to being male during their life cycle, while others... There is so much variability in how sex is handled in the biological world, to say nothing of plants, which just... Some plant species have a male and a female plant, but many of them are hermaphroditic, where they have both sets of sex organs on them. Some animals are hermaphroditic and have both sets of sex organs. So sex and genetics is very complicated. And we're going to do a whole day on it, but we're not going to do it today. Yeah. You're amazing. I know you've earned a rest. That is the most amazing thing I've ever heard, and you're my hero. Did you share that with me? I have never appreciated a person in my life more than I appreciate you right now.
What did you do?
I should make you a sign, and I should hold it up. And you too, actually, because you've been doing a ton of cleaning too. You two have been amazing, and I appreciate it. Containing about 100,000 genes. Normal cells have 46 chromosomes. 2 copies of each. And when we say 2 copies of each, we said already 1 maternal and 1 paternal. Do I need to define maternal and paternal, or are we comfortable with those terms? Maternal means from your mother, and 1 paternal means from your father, right? 1 from biological mom, 1 from biological dad. So this is the physical basis for Mendel's model, right? So the last thing I'm going to write, last thing, I promise. So when we say Joe is heterozygous for, what did we do yesterday? Hitchhiker's thumb? For a hitchhiker's thumb, yeah, that's true. That would actually be a straight thumb, but that's okay. And I'm not, but regardless, that means this. That inside Joe, sitting here with his notably straight thumb, we could zoom in on his hand and we would see he has a straight thumb, or possibly a weird tumor growing out of his hand. And if we zoomed in on that, we would see, of course, that it is made of cells, right? Right? And if we zoom in on one of those, we would see that the cell contains a nucleus, and inside the nucleus is a bunch of DNA, right? Is that cool? If we zoom in on that, that's right, guys. Yeah, listen. If we zoom in on that, it actually means here's the nucleus, right? And we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. We have 26 chromosomes. Is everyone comfortable if I just draw that many? Is that cool? What it basically means is that right here, there are 2 genes, right? And so I'm going to zoom in on that, right? And what that means is that we have one that goes like A, G, C, C, G, C, T, ... Another that goes A, G, 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 A, C, T. And this, and again, I want to be really clear. I'm not claiming that this is the exact mutation. I don't know the exact mutation. But do you get the idea that, like, there's 2 different codes, right? And this one here might be the big T, and that one there might be the little t. Does that make sense? So it's 2 different codes on the same gene, from the maternal and paternal chromosome. Does that kind of zooming out to zooming in make sense? This is how we talk about phenotype. My thumb is shaped a certain way, and for the purpose of this, we're going to pretend I have a straight thumb. Right? So there it is. That means we actually zoom in, and somewhere in my cells, it means that I have 2 copies of this gene, which means there's 2 chromosomes in every one of my cells, and one of them has an instruction that says, hey, we'll teach you how to build the protein that builds the muscles in the thumb. And it's A, G, C, C, G, C, T, blah, blah, blah. It keeps going for 100,000. And then my other copy says, oh, actually, it's A, G, 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 A, C, T, keeps on going. And those 2 are different. And it turns out that one of those is dominant over another. But what we're really talking about is the code of that. And the last thing I want to say is we can actually look, the entire human genome has been mapped, which means, like, if we want to see the gene responsible for hitchhiker's thumb, we can look that up, right? And it is the bendy thumb gene. Well, that's great. I want to know the actual name of the gene, though. Give me the name. There it is. There it is. The gene responsible is the diastrophic dysplasia sulfate transporter gene. All right? That's a gene that humans have. SLC6A2 is the gene. All right? And if we look this up, I want to know the sequence. Alleles sequence database. No, I do not want to donate to you. Okay, so right here, if you look, this is the gene that we're talking about. And if you actually look here, it tells you its chromosomal coordinates. If you want to get to this gene, no problem. Look here, it tells you right where it is. What chromosome is this gene on? Look there, can we see? It's on chromosome number 5. Again, how many chromosomes do you have? Yeah, so if you look here, human chromosomes, we have the 46, but they come in pairs, right? So we have 1 through 23. This one's on chromosome number 5. Chromosome number 5. And how would you count in? You count in starting at the 149,960,758th base pair. So the 149,960th, 758th letter. And then you count on until the 149,987,400th base pair, which means that this is about 27,000 letters long. That's the code to build this little gene that controls, amongst other things, ends up resulting in your thumb.
People that count these letters individually figure this out?
Yeah. The entire human genome has been mapped. And I'm wondering if we can actually see the sequence.
Yeah.
It's verifying I'm human. Oh no. Because you can actually find gene sequences. Usually I look in advance so that I can pull these up because I'm not as good at bioinformatics as I used to be. I'm not up to date on all the best. If it was the protein structure we wanted, I'd know where to go. You thought wrong.
By the way, why can't some people not stick up their
The ring finger. I can't.
I can't. That's as far as I get.
Yeah. Well, the short answer is that you actually, to do so requires a lot of muscle coordination because your ring finger has no muscle of its own. It shares a muscle with the middle finger and shares a muscle with so the muscles that control your fingers are actually way down here, right? So they're connected via tendons, but the muscles that control your fingers are here. So you can feel as you flex your fingers, it's muscles in your forearm that are moving, and this one doesn't have its own. So for you to move just this finger, you actually have to use a few muscles at once. You have to use the muscle that wants to pull these two down and the one that wants to pull this one back up. Isn't that weird? I won't bore you with me trying to find the sequence. I'll get it for you tomorrow. But this is, this shows you all of the base pairs, and you can actually look up, I want to find the actual letters, but you see here, it's actually showing you in this gene where there are differences between us and a bunch of other species. And you can see where we have similarities and differences to other species. I'll look that up later, but yeah. Anyway, that's all for this note. You guys are good to go. Yeah, one sec. I do want to see.
So weird, and it hurts.
I know it's weird, eh? Well, we'll look that up anyway. So I'll put this back up. That's it for today. But I want you to keep that in mind. All that we've covered today. Go back where? Here? Cool. Yeah. Yeah, you bet. What? Don't say that. That's an awful thing to say. Why would you say that?
This whole time I thought you were ignoring me. Muscles are weird things.
Muscles are very weird. But at the end, the muscle is just a big protein that does one thing. When exposed to an electrical signal, it shortens. That's it. All muscles are. Simple proteins, actually. There's actually a bunch of proteins moving against each other called actin and myosin. And they just go and shorten. And when Riley's done with this section, you can just go move it yourself. Find what you need.
Even more than the other one.
Let's make it go to the actual person. So bad. Degrees, not inverse degrees.
By the way, guys, I'm about halfway through editing your paper submissions. I hope to have them back to you sooner. Boy, it's taken me time. It's taken me a long time for paper. So I'll get those back to you all at once. The dichotomous keys should be back to you if you look at your grade checker, which I did post a link to. You can see how you did on it. Your physical ones are back there if you get to see physically. They're well done. Very pleased. I learned so much about animals. I learned a lot of it, mostly animals. Some people did other things. Mostly animals. Few plants. But I really enjoyed them. It was a very well done setup.
No, I only studied
Yes. It was so good.
I love them. They're awesome.
Sorry. Why is she ignoring me? And then just never ever ignore me. I'm so You asked the wrong person.
I was so
Turn the lights on. I'm finding it very dreary.
No way, you're the person.
I'm finding it very dreary. Yeah, let's open some windows. Oh gosh, that's somehow not less dreary, is it? I think it's nice and loud. Would that be nice? Yeah.
I tried to. Sir J.
There we go. That's fine. Okay. We're not made that in grade 9. Grade 9 electronics. Because I like it. Oh, I doubt it. Let me try. Oh, that's interesting. Show me. Yeah, that's like, wow, that one will really go though. Like mine. Wow.
You know what? We need to just why
I can't believe I did that.
I'm always interested that my hands are my daughter has weight as immediately smaller hands, and I think like probably a good half a centimeter shorter. Like she can spread to like 3 keys further on the piano. My hands just do not spread out. It's very frustrating.
I'm scared. I'm doing a salsa. Just try.
Okay.
Miss Orlando.
That was an
How
What's going on?
where's the water
Well,
bottle?
listen, guys, I don't teach grade 5, and I don't teach grade 5 for a reason. Alright?
Yeah.
Every activity is stopped.
Exactly. What happens if it's simple cup? Full on water.
No, it's because you hit my head.
Got water all over.
Since you're at that interval there, tritone. It's such a nasty interval, right? Like it's so.
Sounds like it's for like your alarm system.
It is upsetting, right? Well, it is, yeah. So the symptoms is, and then like that. That ugly nose. Right? Like it's such a. It's really like that song should go. Nope. Right? But it has that ugly tone in there that makes it so interesting, right? There's ugly tones in the back. The witch? I didn't know.
Yeah, so much to do what I asked you for right tonight.
Well, you think like what other thing could you do that with, right? Like if you did like instead, would be like right? Like say you do that in Star Wars, all of a sudden it's like the most revolting. It goes from sounding adventurous to sounding like horrifying, right? Like,
I just sent it to
instead, right? Off, or like Superman.
What the hell is that?
But if it was
Is that
not heroic, horrible, ugly, yes. Oh, Oh, I didn't have to.
Yeah.
I don't actually.
It's good. We'll see.
No, like, no.
You buy a shawl. It's I'll smoothly inside the song. I want to keep that.
The royal. I'm not a great coach.
Now I feel bad that you literally like put your water all over the place.
Yeah, Chris.
Like what the heck?
How are we doing? Yeah. Congratulations, that's exciting. Are you excited?
Yeah. My only fear is that
Probably gonna go to night roll and then doing transfer.
Are you?
Yeah.
Why?
Or
You're great.
I'm not good at it.
Have you ever driven?
Yeah, I'm practicing.
Okay.
Driving scary,
Yeah, no, I just don't trust it.
especially when there's other cars. Be scared.
Not for you, right?
Driving is really scary. Beware.
9 out of 10 for the questions.
Wait, did you do it?
Yeah, you'll find it online.
Oh, online?
Yours is great, by the way. Very good, right?
Thank you.
Now let's check your rubric. Let's see what
Like,
happened
I
to
was
your 3%.
looking at it on the green here, and I was like, I have no idea what he's talking about.
Let me take a look. Because I liked yours.